Okay guys, so today we're going to review cell transport, which is basically how things move in and out of a cell. First thing we're going to have to review is the cell membrane. So materials move in and out of the cell depending on two factors. Number one, interactions with the cell membrane, and two, concentration levels of the materials that are trying to get in or leave the cell. Interactions with the cell membrane. The cell membrane is in charge of controlling what enters and exits the cell. Its specialized structure allows it to be semi-permeable, meaning only certain molecules can cross. So if you look at this picture of the cell membrane, okay, this cell membrane is semi-permeable, which means that some things will be able to go through and others won't be. An example you can think of is a fence, like a wire fence. Some things can go through if they're small enough, but the larger items or larger things won't be able to go through because they won't be able to fit through the openings. Similar to the cell membrane. Cell membrane is semi-permeable. The structure of a cell membrane consists of a phospholipid bilayer, which is formed by two layers of lipids. If you look at the picture, okay, these little tails would be the chains of carbons that you find in lipids, and you have two layers. So there's your phospholipid bilayer, bi meaning two. We also have embedded proteins, which are going to serve as channels or pumps for molecules. So these purple things on the membrane or in the membrane are going to be your proteins. Carbohydrates, which serve as signaling molecules for recognition on the outside of the cell. So here you go right here. These are the carbohydrates that are going to help signal molecules. And then the cholesterol, which makes the membrane fluid, so it's not just rigid. And this picture doesn't show it, but they would be embedded similar to the proteins. Okay, so concentration levels of material. A concentration gradient is the difference in concentration of a substance between two locations. If you are moving with the concentration gradient, you are going from high to low concentrations, which cause a substance to return back to homeostasis. This process doesn't need energy and is known as passive transport. So if you have too much of something in one area and a little bit in another, they naturally want to go to the molecules, or whatever you have wants to naturally go to where there's less, so that it can even out, so that it can be equal. So that's what this is, going with the concentration gradient. You can also think of this picture of this person riding a bicycle from the high point of a hill to the low point of a hill. So he's not going to need any energy to do that. He's just going to have to basically stay on top of the bicycle and go down the hill. So from high to low, no energy. So there's three types of passive transports. We have osmosis and diffusion. Diffusion is going to be the movement of a substance from high to low concentration. And osmosis is the movement of water from high to low concentration. So notice how they're exactly the same definitions, except diffusion is moving a substance. It could be anything. Osmosis is the movement of water. So osmosis is specific for water. And here you can look at a picture of osmosis happening. Water is going from high concentration here on the left and to low concentration on the right. And then diffusion is over here. Here, this isn't water. These purple circles are going to be, whatever substance, and it's going from high concentration to low concentration. And the last one would be facilitated diffusion, which is the movement of a substance from high to low concentration. Again, same definition as diffusion, except this one is using a protein channel in the cell membrane. So these are going to need a little bit of help. And they're going to be using a protein channel to go from high concentration to low concentration. Okay, now moving against the concentration gradient. means that you're going from low to high concentrations, which causes a substance to move away from homeostasis. This process requires ATP or energy and is known as active transport. So in this case, think of a person trying to go from the low point of a hill to the high point of a hill. On a bicycle, it's not going to be easy. That person's going to have to pedal hard to go up, and they're going to have to use a lot of energy. So think about going from the low point to the high point. You need energy. Similar here, when you go from low concentration to high concentration, you're going to require energy because the molecules or whatever it is doesn't want to naturally go in that direction, so you have to force it using energy. There's three types of active transport. You have protein pumps, endocytosis, and exocytosis. In a protein pump, you're going to move a substance from low concentration to high concentration using protein pumps and energy. So the energy is going to come to the protein pump here and is going to grab the molecule and force it to go to where there's a high concentration. Endocytosis, think of endo and think of the word enter. So here there's going to be a movement of large substances from low concentration to high concentration into the cell. It's going to enter the cell. So this top picture is showing endocytosis. And then exocytosis is the movement of large substances from low concentration to high concentration out of the cell. Think of exocytosis, exit. So the bottom picture here is showing the exocytosis happening. Cells can be in different solutions. Cells always strive to maintain homeostasis by regulating their concentrations in an isotonic solution. The cell is at homeostasis. So cells want to be at an isotonic solution, where the water is going in and out at the same rate. So the shape of the cell is not going to change. It's going to stay the same. So this middle picture, you can see how this blood cell and this plant cell, the water's coming in and leaving at the same rate. It is happy. Nothing's happening to the cells. They're staying the same. In a hypertonic solution, there's a lot of solute outside of the cell, so there's a relatively high concentration of water compared to the inside of the cell. This causes water to leave the cell, and the cell is going to shrink. A salty solution is a hypertonic solution. So for example here, if you have lots of salt on the outside of the cell, then that means that you have more water on the inside. So water wants to go from high to low, so the water is going to leave the inside of the cell, and it's going to result in the cell shrinking. If the cell is in a hypotonic solution, there's less solute outside of the cell, so there's a relatively low concentration of water inside the cell compared to the outside. Low solute on the outside means there's lots of water. So this is going to cause the water to go into the cell, enter the cell. In an animal cell, this can result in the cell bursting or lysing. In a plant cell, the hypotonic solution creates a turgor pressure, which causes the plant to stand tall. So that's why plants stand tall, because the water is coming into the cell, causing the cells to fill up. Alright, so those are our top 10 for this section. Good luck on Friday's quiz.